Okay, so I will start. My name is Laura Levy, and today I will talk about genetic diversity and comparative genomics across Leishmania Viannia species. So first, let's talk about a little bit of the parasite. So this parasite belongs to the subgenus Kinetoplastida and the family Trypanosomatidae. Also, this parasite is transmitted by sandflies and causes an important disease that is called leishmaniasis. The Leishmania genus includes approximately 20 species associated with human infections, and according to the parasite developmental pattern in the sandfly vector, are divided into subgenera. So we have the Leishmania subgenera that includes species as Leishmania mexicana, amazonensis, donornavi, infantum, and others, and Leishmania Viannia subgenera that include species as Leishmania brasiliensis, panamensis, guyanensis, and others. So the Leishmania haplogenome is 33 megabases in size and contains about 8,000 coding genes. It is organized into 36 chromosomes in species of the subgenus Leishmania and 35 in those of the subgenus Viannia. Chromosome size varies from 0.3 to 2.8 megabases. And here we can see a geographical distribution by department of Leishmania species reported in Colombia. Leishmaniasis in Colombia shows variation across departments, and the most notable species within this subgenus include Leishmania brasiliensis, panamensis, and guyanensis. For a deeper understanding of the genetic diversity of this parasite, we built a database of genomic variability of Leishmania viannia in Colombia to analyze population structure and genome evolution. So 65 Leishmania Colombian isolates were sequenced using Illumina whole genome sequencing, and we had 186 sequence samples with publicly available Leishmania Illumina datasets. We got a database of the genomic variability of about 3 million SNPs. And from these SNPs, we got this result, the maximum likelihood phylogeny. And the important thing to say here is that we can see two large groups, the Leishmania brasiliensis and peruviana group, and the Leishmania guyanensis and panamensis group. And to further analyze the structure within Leishmania viannia populations, the two main species complex were analyzed separately, following population genomic approach as admixture analysis and neighbor joining tree. Here are the results of the sample clustering obtained for the Leishmania brasiliensis and peruviana complex. Here is the admixture result, and here we can see the neighbor joining tree result. So in the admixture result from K2 to K4, the Leishmania peruviana isolates are shown in red, and therefore the Leishmania peruviana populations can be differentiated from K5 as previously reported. The Brazilian isolates, LBR1, can be observed in green from K3 onward. The Peruvian isolates of Leishmania brasiliensis population LBR4 can be identified from K4 onward. Here we have two important populations, the Orinoco-Amazon region isolates and the Andean region isolates. And the differentiation of these two populations we can see from K6 onward. The Leishmania brasiliensis peruviana hybrids are shown as such throughout the entire analysis. So we can see here that we can see two colors, two pattern colors, and this means the hybrids isolates. And here we can see the results for the sample clustering obtained for the Leishmania panamensis and guyanensis complex. The Leishmania guyanensis appears as distinct group from K2, later splitting in two subgroups at K8 and K10, separating the Colombian and the non-Colombian isolates. A second group of 4 guyanensis isolates forms a group from K4. Isolates from Panama form a cluster differentiated from the Colombian isolates from K3. And here we have that the Colombian isolates were separated in 3 clusters. Are these ones? LATAM 2, 3, and 4. These ones are the Colombian isolates, and these ones are the isolates from the Eastern Andean mountain chain. And these clusters are evident from K5 to K6, from K5 and 6 onward. Given the observed diversity and differentiation of Leishmania viannia populations in Colombia, we decided to sequence the complete genome of some representative isolates. So 10 Colombian isolates were sequenced using a long-read technology. The haploid genomes were assembled into 43 to 100 contigs per assembly, with an average of N50 of 975 case-base pairs. The average genome size was 34 megabase pairs, which was more than 2 megabase pairs longer than the size of the current reference genome assemblies. The GC content ranged between 57.6 and 57.9, that was close to the GC content of the reference. When contigs were assigned to chromosomes, each contig level assembly was highly contiguous, having a range of 24 to 31 chromosomes reconstructed in one single contig. This graphic shows 3 arcs. These are the result of the genome's assembly. Shows here the Leishmania brasiliensis circus, the Leishmania guyanensis circus, and the Leishmania panamensis circus. And I will say that the principal results of this study is that Leishmania viannia species genomes are highly conserved, but are differentiated by multi-copy gene families. The gaps observed can be related. So we can see here gaps in chromosome 2, in chromosome 20, and chromosome 34. And the gaps observed can be related to the repetitive regions, of which could not be resolved with the nanopore reads. About the gene count and the repetitive regions, overall gene count across these assemblies range to 9,172 to 10,242, an increase over current reference genomes. So what we can see here is the alignment among the 3 representative genomes are here, and we compare these genomes to the 2 reference that are Leishmania brasiliensis and Leishmania panamensis. The pangenome reveals variation in gene presence and absence across the species. So we have the result of 8,749 orthogroups. And also this pangenome allows us to identify the core genome, the species-specific genes that corresponds to the accessory genome, and the multi-copy genes. The core genome is containing about 6,635 orthogroups and also 4,932 single copy genes, 35 duplicated genes, and the remaining had 3 or more copies per genome. The size of this genome corresponds to nearly 60% of the genome, supporting the stability of the gene content across the species in terms of functionality. We found gene families with increased copies in new assemblies, such as amastin surface glycoproteins. That increased from 51 to 190 copies, primarily located on chromosome 20. And what we can see here is a principal component analysis of the number of copies in multi-copy orthogroups. So in blue, we can see the Leishmania brasiliensis species. In green, we can see the Leishmania guyanensis species. And in red, we can see the green Leishmania guyanensis and red Leishmania panamensis species. So the cones successfully differentiated species within the Leishmania Viannia subgenus, but except for the older reference genomes, which had fewer recovered gene copies. And that's why we see here a Leishmania panamensis isolate clustering with the Leishmania guyanensis. Excluding reference genomes, 35 multi-copy orthogroups show difference between Leishmania brasiliensis and Leishmania panamensis guyanensis complex, including notable genome families. So here are the 35 multi-copy orthogroups, and this corresponds to the different species. And as we can see, there are different multi-copy of gene families shown here. To take home bioinformatics in the file of bioinformatics, we have assemblers, error corrections, genome comparison, and pangenome construction. And in the file of the molecular biology, we have a low-cost sequencing with a high-quality reads. But what's next? Detailed study of gene families, the molecular markers are important to study, and more genomes with higher resolution. Thank you to the Universidad Nacional, Universidad de Antioquia, el Centro Dermatológico Federico Lleras, MinCiencias, and ExaCore and GenCore from the University of Los Andes. And thank you.
Thank you very much, Laura, for a beautiful talk. Any questions from the audience? Can you approach here to make the questions? We don't have another microphone. Thank you.
Hello.
Hello.
Excellent work. Congratulations. Have you looked at the kinetoplastic DNA? And do you know if there are differences among these species at that level? And then the other is, can you speculate where in the genome are differences that are responsible for phenotypic differences? Like I'm from Peru. I'm always been curious about Peruviana versus Brasiliensis and how one causes mucosal lesions and the other doesn't. So I don't know if you can speculate on where are the responsible regions for those differences.
Okay, the question was very long, so please. The first question was about kinetoplastics?
Yes.
But I don't understand the question.
If you sequence the kinetoplastic DNA and if you see differences, species-specific differences at the kinetoplastic level.
We sequenced the kinetoplastic DNA, but we didn't see a difference between the species in terms of the kinetoplastics.
Okay. Thank you.
And the other question?
The other was just speculation on whether these regions of variation are responsible for different pathogenic properties.
Okay, so that's why we are trying to investigate. Now, we know that there are structural variants, but we don't know now if they cause some phenotypic special like symptom in the disease.
Listo. Gracias.
Okay.
Thank you. I have a question, actually three, but we don't have time.
Okay.
I will try to do very briefly. The first one, did you evaluate the genomic variability of treatment resistance genes? No.
No, what we do was like, well, we have our genome and we look up for genes. We have like a lot of genes, but we don't like look up for specifically resistant genes.
Yeah, perfect. And the other two questions are very short one. The long read sequence was with nanopore, right?
Yes.
And did you multiplex the samples, the isolates?
Sorry?
Did you multiplex the samples? Or it was an independent sequencing run, or all the isolates you put together in the same flow cell?
We multiplexing the samples.
Yes, and the last question, how many genomes did you use in the pangenome analysis?
In the pangenome okay, we use the sorry, pangenome analysis. So for the pangenome analysis, we use the I forgot. Sorry.
No problem at all. Thank you very much. Congratulations for the